I think no more waiting. So good evening, everyone. This is Dr. Mahesh Veluswamy here, and I thank everyone on behalf of LLB School and CPMB&B for being here with us today. And today it's our pleasure to host Dr. Arun K. Shanker, a very special guest from Central Research Institute for Trail and Agriculture, Krida, Hyderabad. And rather than me giving brief introduction about him, I request Dr. Divya Sri to give an introduction about him because as they know each other very well. So Dr. Divya Sri, I request to give a welcome message and give something about his research and talk something his research background. Thank you.
Hello everyone, I am Divyashree Nageshwaran and a PhD from University of Cambridge. Most of all, a TNAU alumni. And to say a few words about Dr. Arun Shankar, he is a plant physiologist come bioinformatician, and most of all he's also a TNAU alumni, and he's going to talk about the recent work on that is published in the New Journal of Chemistry, titled on Whole Genome Sequence Analysis and Homology Modeling of Main Protease and Non-Structural Protein 3 of the SARS-CoV-2, which reveals an azapeptide and a lead inhibitor with possible antiviral properties. So today he will share with us his published work, and it is also another example from following yesterday's talk, that it's an interesting example of that it's a transition from plants to identifying antiviral compounds against the COVID virus. So it's another example of research from a plant physiologist in relation to SARS-CoV-2. So thank you, sir, for accepting this talk, and yes, you can take on from now.
So before handling the session, thank you so much, Dr. Divya. Before moving on, I would like to give some important technical details for all the participants. So here are some technical notes. So for the participants who is using WebEx, please kindly turn off your webcam and mute your audio for better webinar session. And participants from both YouTube and webinar, try to note down all your questions and we will do our best to address all the questions at the end of the session. But Dr. Shanker already mentioned that if there is some necessary in this time, we allow some important question during the, like every 10 minutes we can give opportunity to people for important question. And for the participant in webinar, you will be always in mute mode. So during the question session, please use your raise hand option, or else you can message in the comment box. And last but not least, don't forget to send your feedback via this Google form. So that will be really helpful for us to make really quality of lectures in future sessions. So with that, I request everyone to give full attention to Dr. Arun K. Shankar and help me in hosting with the wonderful session. So thank you so much. And Dr. Arun K. Shankar, thank you so much for your patience, and I'll let you take over the session. Thank you.
First of all, thank you very much for the LLB host and Divya Shri, and also TNAU, CPMB, and the organizers, everybody, for having called me for this talk. It's really a great pleasure to address you people, and especially because I'm associated with TNAU, it's a matter of pride to involve myself with something with TNAU. And firstly, I want to tell you that I'm a plant physiologist, basically. I'm not a virologist or a drug discovery or something like that. But the whole study is about, it started off with a small intuitive study. It was like, if you remember, the whole thing started, the outbreak started somewhere around January 15th. And we work on NICRA. There's a NICRA project of ICR. I am a PI of a project where we do computational genome analysis and gene mining in maize. Basically, we try to find out genes and candidate genes for drought tolerance and other abiotic stresses, heat, drought tolerance, like that. And that is the project I work on. And my project staff, that is a bioinformatician, also works on this. So what we did was, what we thought of was, when the genome of this virus started to come in NCBI, we just thought that why not we do some practice on analyzing this genome and seeing how it is related to the other viruses, known viruses and sequences and all that. It was just a thought that struck my mind, and I asked my project staff, Divya, Banu, to download the genomes and start doing some analysis. And that was how it started off. That then, another thing I'll tell you is, the whole study is a very simple study. There is nothing very special in it. We used very, very simple bioinformatics approaches, very accessible and open access server-based analysis we did. Everything from homology modeling to protein ligand interactions, and also docking studies. Everything was server based and very not computationally very intensive. It was all simple things. Only thing is, we started off very early, in the sense that there were only 6 genomes of the virus available when we started off, and also there was no knowledge about the basis of the virus. The virus was not even named at that time. In fact, we didn't know that it was related to the earlier SARS. And that was when we stuck up our pay dirt. Like, we saw that most of the homology modeling led us to proteins which were clearly related to the earlier SARS. And immediately what we did was, we put it up in a preprint server. So people who are listening to this lecture, and the younger people, like younger researchers, I would like to tell you that preprint servers are not very popular with biological researchers. It's very popular with physical sciences. But here, let me tell you that if you find something interesting and find something new and novel, I would suggest that you put it up in a preprint server, so that you can maintain your priority. That is, prior art is maintained. So that is what we did. Simple study it is. So now I'll get into the study as such, proper study. This is actually, like you can see in the title, it's a whole genome analysis of this virus, SARS-CoV-2. And we did some homology modeling of the main protease and non-structural protein 3. And we found out 2 compounds, that is an azapeptide and a lead inhibitor with possible antiviral properties. And our team is Divya, Banu, Anjani Alluri, and Samriddhi Gupta. All of us from different places. Actually, Divya Banu is my project staff, Anjani is my student, and Samriddhi Gupta is from the School of Life Sciences, Department of Biochemistry in University of Hyderabad. So what we did was, and we actually today, as a matter of fact, I would like to share that today is when our final paper has come online in New Journal of Chemistry. This is the publication. It has come out today. I'll share the URL with you if you want the full text. Now, here is the hypothesis. Actually, what we did was, we actually hypothesized that this virus can have homology with several proteins of the earlier SARS. We thought that there must be definitely a high degree of homology. So we saw that these proteins, the PL-PRO, that is a papain-like protease, and 3CL-PRO, that is 3CL-like protease. These have a very important function of processing the viral polyprotein. Polyprotein is a large protein of the virus, which is further cleaved into several other NSPs, or non-structural proteins. So this was the basis of our starting off with the study. That is, the hypothesis was that. And this PL-PRO is to be noted is that it performs a function of stripping the ubiquitin in the host. That is, it facilitates the virus to replicate, and it also facilitates the virus to evade the immune response. This is what I want you to note. It helps the virus evade the immune response from the host. So that is a very important protein. That is a very important target for drugs, because once we target that successfully, we can not only induce immunoprotectant in the host, we can also stop the viral replication. So it's a dual sort of approach if we go for PL-PRO. And they also have other roles of disrupting signal cascades in infecting cells and protecting the uninfected cells. So these 2 proteins, which form the main protease, which are in the main protease, are very good targets. So with this hypothesis, we started off. And actually, to go into, I think this probably most of you would know, must have read about the coronavirus. It is basically a zoonotic. It has got zoonotic properties. It jumps from species to species. It has been jumping from bats to humans, camels to humans. The SARS, which was earlier found, was 2002 in Guangdong province. That was the first of this. Although there are more coronaviruses that actually affect the humans, there are 7 in fact. These 3 form the main ones. That is the SARS, the MERS, and the present one, that is the SARS-CoV-2, that is taken a pandemic form now. Now, now going into the study, Ash, I would want to just briefly tell you about the genome structure of the virus. The virus is, actually has a 27 to about 30 kilobase genome size. It is quite a large size for a virus. Most viruses don't have this size. This is quite a large size. So, and they are especially this viruses, like coronavirus in general, are positive sense RNA viruses. That is, it is already a processed mRNA. Once it gets into the cell, host cell, it directly starts to transcribe. That the protein starts to transcribe. There is no DNA to RNA, or there is no reverse transcriptase like we have in HIV and all that. It is so this is a very adapted advantage for the virus. It is a positive sense RNA. It gets into the host and immediately starts its work. And this has 2 main ORFs. That is one ORF 1A and one ORF 1B. There are several other ORFs, but these are the main ORFs. And these ORFs actually correspond for polyproteins that are processed, and 16 non-structural proteins are processed from these polyproteins. And the remaining portion has got a structural protein. That is, these are the non-structural proteins. That is 16 non-structural proteins. And they have, it also has a structural protein, which is a spike protein. You know the spikes that you must have seen in the photographs or pictures of this virus, the spikes that jut out of the virus. That is a spike protein. There is an envelope protein, there is a membrane protein, and a nuclear protein. This is the basic structure in nutshell of this virus. And it is also known that this can undergo, one important thing of this virus is it can undergo recombination and in the genome within the host cells, and it can recombine with other coronaviruses. That is how probably it evolved. The origin is actually a big question mark. We don't know how actually it came. We don't know the intermediate animal also. So, but it has got a recombination factor, that it has got a high rate of recombination, and this is one of the important aspects by which it adapts itself to new hosts. That is the new receptor cells and all that. So these factors give it a very high adaptive advantage and the capability to jump across species. Now, this is the genome structure. Let me tell you, like I told you before, this has got a about 30,000 BPS genome structure, and the ORFs, like I mentioned, ORF 1A, 1B, and several ORFs of ORF 2 to ORF 10. 1A and 1B are important for the non-structural proteins, which we'll be discussing. So this 1A and 1B almost forms the about 20,000 of the base pairs. The rest of it is, this 10,000 forms the rest of the base ORFs, which is 8 ORFs. So in this is actually, I have taken this genome structure from a study where the authors have actually done discovery of potential drugs by computational methods. That correctly corresponds to what study I have done. And these things that is mentioned here, that is these are all the PDB entries of the particular protein. Like for example, it is NSP5, the main protein which I talked about. NSP5 and NSP3 is where we dealt with. NSP5 is the main protein. This is the PDB entry of NSP5, and NSP3 is over here. NSP3 is here, and the PDB entry is 36A. So these 2 proteins is where we targeted. So the rest of the proteins, like I told you in the previous slide, the spike protein, the envelope protein, the membrane protein, and the nuclear protein. Here, now coming to the main aspect of our study, like how did we go about and what did we want to do? To tell you the truth, we didn't go on starting off to find an antiviral or a vaccine or something like that. We just wanted to analyze the genome. We wanted to see if there is a homology with other viruses. We wanted to see what this virus was. That was the basic objective. That was the basic intuition behind the study. But when we kept going, we saw that the NSP3, that is non-structural protein 3, I'm sorry, one second. Yeah, this non-structural protein 3 here, this thing, and the CL3 probe, both were having a very high degree of homology with the previous SARS. That is where when we did online homology, we saw that about 50 templates matched with them, and the top templates were exactly almost very highly identical, 96%, 98%, like that with the previous SARS. So at that time, the virus was not named SARS-2. And the papers were coming out. This was about February 4th or something like that. February 4th it was. So papers were coming out in bioarchives and chem-archives, and people started to speculate on how this virus is related to the previous SARS. That is when we put up our paper, and then we put up our paper saying that the CL Pro and PL Pro were closely related to the earlier SARS. So this is how we went about. And to know, tell you about these two proteins, that is NSP3 and NSP5. They are involved in the processing of viral polyproteins, like I told you before, and they actually split the viral protein into several NSPs and package it into new generation viruses. This is the main function. So this is why these two proteins can be very good targets for drug development. Then the computational analysis we did was, basically we started off with the homology modeling. Like I told you, we did it with known templates. We did it with Swiss model servers. That is a very automated sort of homology modeling, not much of a manual modeler and all. We did not use that. We just uploaded our sequences, and we did a homology model, and we got back query results from Swiss models from several templates. We started analyzing the templates for similarity and also identity. And we saw several templates. Like I said, we saw several templates. We just, when we looked through the template, that is when we saw that certain templates, that is one or two templates, had ligand complexes. That is where we narrowed down. We omitted the templates which did not have ligand complexes. That is, most of the templates already in the PDB database were just the crystal structure of the earlier SARS. Earlier SARS had been worked very well, and several other coronavirus had been worked very well. So PDB had a lot of entries of similarity with this protein. But what we did was we narrowed down on homology models, templates which had ligands with them. And some of the ligands were not actually antiviral ligands. So we further narrowed down to ligands from literature which had antiviral properties. So that is what we thought, that the viral replication, that is, we further narrowed down to ligands which could possibly stop viral replication by inhibiting these proteins. So that was the idea. And this is where this is the...
Hello, sir. Sorry for the interruption. I just want to know why the filtration criteria has, the structure has ligands. Why you kept as a filtration criteria?
Actually, why? Because, you know, when we started to get the templates, that is when we thought that we can look for ligands which can have antiviral properties. Otherwise, you know, getting a template without just a template which has similarity with the SARS, we only know the tertiary structure of the M protein or PL Pro. That is already known. So that wouldn't form a discovery or a practical applicability. So when we thought that, when we saw that there are templates with ligands and those ligands had antiviral properties, that is when we thought that this could have practical applicability. That is where we narrowed down to those ligands.
Okay, thank you, sir.
This is the pictorial description of how we did it, actually a graphical description. That is the two candidates we got, GRL0617 and AZP, that is azapeptides. And the targets were these. Like you can see, we narrowed down to the amino acid sequences of the targets. That is between 315 amino acid in the frame 2 and a 306 amino acid in the frame 2 between these residues. So here we saw that one had 82% similarity with PL Pro, and that had a ligand GRL0617. And the other had a 96% similarity with C3L Pro, and that was AZP. This is how we narrowed down on the two particular antiviral targets. I'm sorry, antiviral drugs. Now we did a close alignment with the Clustal, and here you can see this is the query sequence. This is the query sequence. This is the template. Same, the other model, again, this is the query sequence, this is the template. There was a 1-to-1 match. That is, almost every amino acid was matched in this particular area. That is, there was a 306 amino acid match in the C3L Pro and a 317 amino acid match in the PL Pro protein. Now, what we did was we took it as a starting point for the drug design. Actually, we didn't actually design drug design de novo. We did not do de novo drug designing. We just matched it with the earlier literature. So here, then what we did was, now this, we had to see if there is a protein ligand interaction with our model as we could see in the template. This is the model we developed. Here is the ligand attached to the C3L Pro. C3L Pro, that is of the main protease. This is the azapeptide. This is our model, which we got from the template. Similarly, this is the other model, PL Pro. This is the GRL 0617 ligand attached to the homology model. Now here, the next step we did was we wanted to compare the binding sites of the model and the template. So here on the right side, this is our model from the SARS-CoV-2. This is the template. We saw in this thing, that is a 3E9S PDB template, which binds to GRL 0617. We saw that almost every amino acid in the binding region matched, except that there was a specific tyrosine here. You can see a tyrosine that was an extra tyrosine in our model. So we had to go further ahead for with Autodoc Vina. Similarly, in the other azapeptide, that is a 2A51, there was a complete 100% match. You must have noted that this azapeptide had a 96% identity with the earlier SARS, whereas the earlier one, that is GRL Pro, had only 82%. This azapeptide had about 12 amino acid differences. So here we did an interaction profile with the protein ligand interaction servers.
Hello, sir. Sorry for the interruption. There are someone who want to ask question. So can we allow him ask a question?
Please do. I'll try my best to answer.
Yeah. Hello, Mr. Lakshman Kumar. Can you ask a question? Hello. Hello, Mr. Lakshman. Okay, I think he's not there. Maybe we can continue like this. Sorry, sir.
Now, yeah, I think he's come or what?
He's not responding, but he.
Okay, then I'll continue.
Okay,
so
okay.
what we did was, next we thought the next step was to find out how well this model, or how well these antiviral compounds, ligands, GRL 061 and 0617 and azapeptide will bind with our homology model. Theoretical chemistry, basically it is. So we went into, Lakshman Kumar has raised a hand. Probably he wants to ask a question now. Stop.
Seems like, okay.
I saw.
Hello, Mr. Lakshman Kumar. Do you have any questions? Okay, I think it might be a mistake, I guess.
Okay, then I'll carry on. I'll continue with my presentation.
Okay.
So what we did was, we went into a protein ligand interaction servers and we did PLIP database. We collected the PLIP data so as to find out the hydrogen bond, hydrophobics, and alkyls, pi and interaction, pi sigma interactions and all. I'm actually not very familiar with this, but my other collaborator, Samriddhi Gupta, had done all this work. She did that in Autodoc Vina, where about 9 conformations of this ligand were run in the Autodoc Vina, and we found out which one of the conformation had the maximum affinity. The affinity of about -7 is an accepted affinity for a good ligand binding. So this we did. You can see that in this GRL 0617 with the PL Pro, there was one which had unfavorable donor, whereas there was a good affinity. Although I must say that in comparison with the earlier SARS, that is reported in literature, and our homology model, there was quite a bit of a difference in affinity. That is, the reported one in literature with the earlier SARS had an affinity of -10 or -10 point something, but we only got about -7.1. -7.1 is, although I must say theoretically, a good affinity and a good possibility of good binding. So here is the next one. The other compound, that is the interaction profile of AZP. AZP had a much better binding affinity than the GRL1, 6017. So here I'll just conclude in a small nutshell. That is basically we had a template to a we had this template from ADB which had 96% identity with the C3-like peptidase, and the compound was azapeptide epoxide, and this had a binding affinity of 7.1. Similarly, the 3E9S of the PDB database of the earlier SARS had 82% identity, and the compound was GRL0617, which had a higher, much better affinity. Although, it must be of minus 7.9 kilocals per mole. To note here was this, which has a lower affinity, had much higher identity in terms of the alignment, that it had a 96%, whereas this one had 82%. So another thing later on, we kept revising our work. As more and more genomes were being sequenced and deposited in NCBI, we did a whole analysis of about 831 genomes of this virus and found that this particular area, that is 306 amino acid sequence and 315 amino acid sequence, was totally conserved. So we can very much say that there is no, this 813 was done on April 24th. That is almost about 2 weeks back. So it can also be said that there's not been any much mutations in this virus. If you have been reading literature, you would probably have noted that there are mutations in this virus which can change the protein structure, like tertiary structure can be changed and binding can be changed because of mutations. We saw that this was not the case. That is these two proteins, that is C3L Pro and PL Pro, and the region, specific region of that is a 315 and 306 amino acid region of that, did not have much of mutations, and they were conserved in 831, and I think probably they are well conserved proteins. And both had, this is what I said, and one of the things from the literature, that these two compounds don't have cytotoxicity to humans. So targeting this part of the genome for antiviral compounds can have a good implication for the development of antiviral compounds for SARS-CoV-2, and it is possible that these ligands we found can be used as antivirals for SARS-CoV-2. And before finishing off, I'd like to tell you from where we got this. That is from our homology models. The main templates we narrowed in on was from the azapeptide was from this paper. It was published in 2005, and a substrate like azapeptide, which inhibited coronavirus. This is a laboratory work done, main peptidase being inhibited by azapeptide. And the next paper is this one, a non-covalent class, papain-like protease inhibitors. That is a GRL ligand. So these two are the main papers from which we drew our templates. This is as much as I can do. I think you must have had some good information from this. Thank you very much.
Thank you very much, sir. And questions?
Should I stop sharing the?
Yes, sir.
But there might be a need, like if somebody has.
Okay, I'll do that. No problem.
So, hello participants, if you have a question, kindly let us know by comment box, or you can use the raise hand option.
So Pranav has a question. Pranav says, what about validation of the models?
Yeah, validation we did with Ramachandran plot and all that. We actually found RMSD of very good validation, high validation. It was very highly significant, both the models. In fact, I have not presented a lot of data here due to constraint of time, and also due to probably I'll be getting into too many slides and too many details. We did good validation. If you want to see the validation data, I can show you now.
Definitely, sir. You can elaborate a bit on that as well.
Yeah, 1 second. I'll have to open my supplementary data to show you the validation. How do I?
So you can
I-
share your screen, sir.
I'm not able to get.
Might be you have minimized the slide.
Yeah, but how to maximize it?
Okay, can you do Alt Tab, sir? Maybe you'll.
Alt Tab.
Yeah.
I'm not able to see your video also.
Okay.
Wait a minute. I can share, but just a minute. 1 minute. Huh?
Yeah, sure.
Yeah, I'll share. I'm getting it. Just a minute.
Yes, sir.
Yeah, I'll go to the validation part of it. Here is. Can you see here?
Yes, sir.
This table is the interacting residue table, and the validation table is here. Yeah, here it is. Can you see the validation table, RMST and raw scores? Actually, I'm not able to see the video. I don't know what I did.
Yeah, to see. Yeah.
No worries. As long as you are able to see, that's fine.
So I can.
Maybe it's a problem with. Let me check it out.
No, it's something to do with my thing. I minimize something. I'm not able to maximize it.
Okay. 1 second, sir. I'll try. Your presenter. Maybe if you send your slides, we can share it from here. But here we can. It's visible for everyone, I guess.
No worries. That's what I'm telling. So long as you can see, I don't have any problem. I'm sharing my Word document now.
Okay.
I'll share you 1 minute. I'll share the. Just a minute. Just a second. Yeah, now I just got the. Yeah, here's the paper, and here is the. 1 second. Here is the. Can you see the statistics of sectional comparison of the templates here?
Yes, sir. Yes.
Yeah, that is the one. And this is the ligand interacting residues. Actually, I didn't go into the phylogenetic tree because now there are more than 3,000 genomes of this virus. When we did, there were only 9 genomes. So there's no meaning in doing phylogenetics now.
That makes sense. That's true.
And this is the structure of the azapeptide, which I told you about. And one important thing I like to tell you is that now is that this thing is this GRL 6012. What we did was we wrote to a lot of labs who work on this virus. Because we couldn't take it forward, to finding something and just leaving it doesn't make any sense. So what we did was we wrote to a lot of labs, and I specifically found some labs which work on this C3L Pro and PL Pro, and we wrote to them saying that we have found this. If you could take it forward in wet work, like laboratory work on in vivo and the virus and all that, it'll be fine. So one of the labs in Germany, Gutenberg University, who's a pioneer in PL Pro, what they did was they worked on this GRL 6017 compound, and they have come up with a preprint now. They've come up with a preprint where they have found that this actually works. They have put it on. It is under review in Nature. So I can even share that paper if you want. 1 second. I'll share the URL later, but I can share that paper if you want. That is a really good thing, to know that what we found out is working in vivo, and it is being probably it'll get published in Nature. That is really good. So we'll have to find that out. 1 minute. I'll open that and show you.
Sir, but at this point of time, do you have the possibility to connect or to proceed with the drug development, collaborating with industries, for example?
Actually, to tell you the truth, like I told, no, I'm in no way connected. I'm in. This is a personal work out of my own interest. I did. So I don't know. I can only communicate my result, and whoever is going to look at it, they are going to do. And I don't actually have any idea of collaboration or anything like that. I don't even have any idea of how to go about these things. Basically, we worked on some bioinformatics things, and like I told you in the beginning, it is a very simple work. Only thing is like the proverbial early bird, early bird gets the worm. We were early on it. We got it early. That's the reason we were able to publish. That's what I think. Otherwise, by now you must see that there are many compounds that have been discovered. Many have been discovered. But this GRL is definitely a promising one.
Definitely. There is a swarming number of papers on the sequence diversity and all those things, and yeah, like as you said, you were the early bird in this research in it. So
which we could do, manage panels. Yeah, tell me, please.
Sir, we have also other question from the participants.
Yeah.
There is in Saranya and Vimal Trivedi. There is a question from Vimal Trivedi. What is the symptoms of COVID-19? Is faster is younger aged or in older age person? I think this is not relevant to the topic. Yeah.
Very general question. So
yeah.
I
Let me tell you something again before like people start to ask questions. I don't know much about the COVID. Actually, you must have seen that COVID is the disease, and SARS-CoV-2 is the virus. We have just worked on the virus genome, and I'm not very much aware of. I think I can't tell you anything about the disease as such because I'm not qualified to do that.
So participants, do you have a questions? You could raise your hand.
I think at this point of time, I would like to and she's also a part of this like this team. And
yeah, she's done a lot of work on this. In fact, she was the one who did all the genome analysis and all that.
yes, sir. And there is another question also on say there are antiviral available for different viruses. There are similarity between COVID-2 to other virus. Why don't you use those antiviral drugs? Since they have better similarity score at protein level as well.
That is exactly what I presented now, because antivirals I have shown is actually working against the earlier SARS. The whole presentation is about that.
Yeah, exactly. Probably this question came from the beginning or so, and Yeah, and it's a double clarity to you, I guess, the person who asked the question. So there's another question from Saranya. Why you have model structure if experimental structure is available?
Sorry? No, actually, I'll tell you one thing. That's what I said. We started off when this virus was not even known. The experimental structure or crystallographic structure of PL Pro is just now done. It is not even published now. And the crystallographic, X-ray crystallographic structure of C3L Pro was somewhere around March when it came up. But we had finished the study and we had even published it in early February 13. At that time, they didn't even know that this had any relationship with SARS.
Yes,
The rate at which the information is coming about this virus is something we should understand. Every day there's work going on.
definitely. And I hope, Dr. Saranya, you got the clarity.
And Divya, can you ask Dr. Shanker to enable his video or slides? Because
Sir, you can, I think, enable your video now, I guess.
My video, actually, I don't see the panel. I don't know what's wrong.
I,
Yeah,
think,
I've done it. Yeah.
okay.
Yeah.
Yes, sir. Thank you.
Yes, sir, thank you.
So
And
I have some question like after the modeling and modeling part, have you like how did you confirm whether this model is perfect? Other than the validation part, have you done any simulation or molecular docking just to confirm the stability of the model?
Yeah. Model stability we did in the sense that we did Autodoc Vina. We actually did Autodoc Vina. I told you in the slides we did ligand protein interaction studies in which
Okay,
we did several confirmations of the ligand, and we could see we also found the theoretical affinities in kilomoles kilocalories.
but if I'm correct, molecular docking studies for having the interactions, finding the interaction, whereas before moving to the molecular docking studies, it's the standard protocol to check the stability of the models. So have you done any molecular dynamic simulation just to confirm whether the model is a stable one?
The model with the ligand interaction or the homology model itself? Which model are you?
The one model that you have chosen as a representative model.
No, the model is actually we did all the validation of the model
Yeah.
in the Swiss model server itself. It
Okay.
had good RMSDs.
Yeah.
The model was because the model basically was developed with a one-to-one match of amino acids with the earlier SARS.
Okay.
So the crystallographic structure is almost the same, but now maybe now since the real model has come out, I mean they have they have come up with the model of the not the homology model, the model the exact model of the PL Pro hasn't come. So we can now there's no point in doing that because already the model has come out.
Okay. But if I'm correct, the Swiss model is only to check the quality of the model. But
that is what we did, not more than more stability.
Okay. Sure. Okay. So, Divya, are there any more questions?
I didn't get it yet. And
Okay.
so why did you choose only 3C Pro and the PL Pro? And did you choose any other main proteins as well, main proteases as well?
No, actually, that is what. See, I told you in the presentation that these are the best drug targets. Okay. I'll get back to where. Can you see my PPT now?
No, sir. Actually, I think it is minimized, I guess.
No. I am sharing something else. No, so I'll share. I'll tell you. I'll answer that question. That's a good question. In fact, that why C3L Pro and the PL Pro of main protease.
And there is another adjacent question to that asked by someone. How did you come to know that those are the only proteases involved as well? So I think that's what you said. Anyway, that is what you like. You found
Now,
that
can you see? Can you see my?
Yes, sir. Yes, sir. Yes, sir.
exactly. Yeah.
So see, we went on this. Okay, this I'll actually give you a slideshow. Now, can you see this performs the one is PL Pro is a very important protein for the virus where it beats the what do you call immune response of the host. Okay, so it helps the virus to overcome the immune response of the host. So attacking that is going to be a very good drug target. That is why we went. That is the hypothesis I told you in the beginning. And also C3L Pro also is involved in the viral replication. So that again, if we attack these two proteins, it's going to be a very good antiviral drug. That is the reason we went for these proteins. In fact, we didn't go. The moment we started to get models, we realized that these are the proteins we should go for.
Did you have any other options as well in the list of templates when you compared?
Yeah, we had a lot of templates. I told you we had a lot of templates which were with. In fact, one of the highest similarity was the RNA-dependent RNA polymerase of the virus. But that is an enzyme, and that had a 99, 98%, 96% similarity, and that was one of the first to be reported. In fact, but that cannot be a drug target as such. That's an enzyme. So that we've reported in the paper. But these are the ones which had ligands, which had antiviral properties.
Yes. And I hope it answered most part of the questions that appeared here.
Yeah, I
And
think.
Pranav has a question.
Structure-based.
Can we tell this approach as a structure-based drug repurposing?
Actually, I don't know if we can say this. This is just what you call, it step by step without based on a simple hypothesis. We struck on this ligands. If you now with the available information, we can do structure-based drug discovery. Now there is much more information available about the virus. Almost everything is has been done in the lab. So at that time when we didn't have any information of the virus except 6 or 7 sequences of the virus. And those were also not complete sequences. Some were only the polyproteins. So this definitely can be done now. A lot of work is being done on structure-based drug discovery of for this virus.
There is another question as well, and somebody asked, maybe they haven't followed or so, but what is the exact role of azapeptide epoxide in SARS-CoV?
Yeah, azapeptide epoxide actually interferes with the C3L Pro, and it inhibits C3L Pro from acting. Actually, C3L Pro is important for the replication of the virus. So azapeptide actually binds with the C3L Pro and inhibits the enzyme. So replication is stopped. So we can stop the replication of the virus. Thus, it is an antiviral.
And then there's another question. Is it able to find out the range of the strains by sequencing the virus samples collected from the infected persons at different locations?
No, this does not fall under the purview of my
domain,
presentation. Yes.
So sorry about that.
No worries.
And I think this answered most part of the topics that were covered. So
we'll look for, we can take 2 more questions if somebody has questions.
Yes. And Pranav, do you have any other question? You can unmute yourself.
Hello, sir. Yeah. So you have done a good work. Now, like, what are the future studies you would like to continue from this work? If any, can you explain a bit?
I'm not going to do any future studies in this because this is something I told in the beginning. It was an exercise we did. My work basically is on maize genome and chlorophyll fluorescence. I'm a plant physiologist, so I may not be going into further into this.
Okay.
But it is interesting to see this transition at times, like doing some in silico analysis with the current situation. So being like a two-in-one scientist between plants and human. And thank you, sir, for this amazing talk, and hope everybody understood the topic. And if you have any other questions, please post it to us. And Mahesh, you can go on.
Yeah, 1 second.
Magesh,
Yes,
are
sir.
you able to?
Yes, sir.
Audible?
Yes,
Yes.
sir. So I would, Dr. Arun K. Shanker, thank you so much for that wonderful session. And now I request to give a vote of thanks from Director of CPMB and Head of Mathematics CPMB. So,
Before vote of thanks, I have a few questions for Arun,
okay,
Dr.
sir.
Arun.
Okay.
Okay. And I think early bird. So congratulations.
Thank you very much.
And I follow all your postings in FB and everything. It is quite interesting. Can you throw some more light on number of sequences that is done in India and how the mutations occur in this COVID? Can you just throw our Indian isolates alone? Because now they are saying more than 30 sequences or 100 genome sequences are already done in India, and how the variations are present. Can you throw some light on that?
More than 100, 200 genomes have been sequenced in India. CSIR is doing the job there. Their laboratory is doing. In fact, one Dr. Vinod is doing it. He's leading the study there, and they are finding some mutations, but they have seen some clades in that. I'm not following that so much, but this is as much as I can share about this. There are more than 200 genomes that have been sequenced. And probably there are some mutations. This virus actually mutates 2 nucleotides per month. So there are mutations definitely, but I don't have any more information to share with this because I've not studied into this.
No. Just I am very skeptical. These mutations have something in your target proteins, and if there is a mutations in that target proteins, whether this ligand and that target interaction may be influenced.
Yes, that is one thing. That is the reason I told you that we did an analysis with 831 genomes of this virus. That was about 2 weeks back, 24th April. In fact, there were more genomes even then. But we took a representative genomes of 831. There was not many mutations in these particular proteins, that is C3L Pro and PL Pro, but there are mutations taking place, and there are speculations, and there are a lot of papers coming in bioRxiv about what these mutations could be. In fact, we ourselves have developed some mutation from GSIH site, that is NextTrain. If you go to NextTrain, there's a site called NextTrain where you can get information about this. There are several mutations in several of these sequence genomes. But I'm not very much aware of what is the functionality of those mutations. Okay.
And also, what is your view on origin of this? Because some view you have expressed, and something because this is for this is for students, just for elementary information they should know. So what kind of this origin? Because you know that SARS COVID-19 alone may be a little bit more we are talking as compared to the other SARS. So what may be the reason for that?
Actually, again, origin, I have been sharing a lot of my opinions and my write-ups on social media. Like you told, you've been following me, probably that is what prompted you to ask this question. But
yes.
Okay. Actually, you know what? Origin, basically, it is from bat. That much as we know. Bat is where it originated. But how it got adaptable, like I told you in my beginning slides, that this has a very high recombination ability. This can recombine a lot. And that is one of the main things by which it can adapt to be zoonotic, that is crossover to from one species to another. And because of the recombinations, and there are speculations that pangolins had these viruses. Pangolins had two or more of this type of SARS viruses, which recombined and came about as a new one. So there are a lot of theories and speculations on this. This thing, we need to know about this. But as far as now the present knowledge is concerned, it has come from bats.
Okay. That's a good information for, especially for students. They have to, as a bioinformatics or biotech, they should know the basics of this, how the recombination occurs. They should follow and then see that how best, because in nature, when you are mingling with the nature, there are so much of things will occur. So like that, they should understand and how to overcome that also. Okay, so as a plant physiologist with the maize genome, so you have a wonderful opportunity to work with this COVID, and as within a short time, I informed to Dr. Divyasri, and she is able to arrange the interaction with you, and TNAU is very much proud of you as well as I. So thank you very much for giving the wonderful piece of your work and recently published that may enlighten the bioinformatics students for doing the further work. Even though they are in plant science, they can contribute very well for various kind of health related aspects, either it is pharmacology or for medical sciences. There are plenty of cross-disciplinary opportunities. Thank you so much. I also thank Dr. Magesh, Dr. Divyasri, as well as your collaborators. Dr. Divya Banu also joined, and I congratulate everybody for being an early bird. We also submit as like that with different targets and with different herbal proteins, one paper to Indian virus disease journal. I hope we will see that maybe in another 1 month it may publish. So thank you so much, Dr. Arun.
Thank you very much for giving me the opportunity here. It's a matter of pride to interact with my alma mater, and wish you all the best in your work as well, sir. Thank you very much.
Yes.
Divya, thank you very much to
convey our regards to Dr. Chitra also. I think who is watching this one.
Chitra was actually the person behind this work, because I was continuously working on this, and she kept on egging me and motivating me for this.
Okay. So, and thank you, Dr. Mohan sir, and thank you so much, Dr. Arun K. Shanker. I hope, can we ask Dr. Nananam also give some vote of thanks? And Divya?
Yes.
Yeah.
Are you?
Yeah, I'm there.
Okay, you can go ahead, ma'am.
Yeah. Very good evening. I profusely thank Dr. Arun Shankar, the speaker of this today's webinar. I know very quickly, as he said, early bird always catches the prey or the worm in the early period. So because of his good work and publication in this corona pandemic period, he has earned peer recognition. It's very nice that appropriately and generously he has shared whatever knowledge he has received during this publication. And additional flavor to this session is he's also our alumni. He's also one of the proud alumni from TNAU. He's a physiology turned bioinformatics. If you see his CV, apart from his expertise in physiology, he has also mentioned about web programming and customized bioinformatics knowledge, and that is how that has helped him to work during this period out of interest, and that has brought great social awareness. I also thank the LLB school partners, Dr. Magesh and Dr. Divyasri, for hosting this exciting lectures in line with the current scenario. Myself and director are really very happy to be part of this webinar, and thanks for the efforts arranging a correct kind of webinar at the right time, at the right platform. I also thank all the participants for showing interest in learning this. Thank you.
Okay, ma'am. Thank you so much. Thank you so much.
Thank you very much, madam. It is really, again, like I told you, it's a pride to interact with you people.
Yes. We are really proud of today, because all are our alumni, and really, the teachers' real feeling is only from.
Okay. Okay.
We thank Dr. Chitra Shankar also for motivating you to do this work. Thank you.
Yeah.
Okay. So now I think it's with all the vote of thanks and nice response from the faculty of TNAU and as well as from students. I think it's time to wind up. So before winding up, once again, we would like to say thanks to Dr. Arun K. Shankar. So thank you so much, sir. So for the participants, please don't forget to give your feedbacks in the form that I have attached in the comment box. So please give your feedback, and we will come up very soon with another interesting lecture next weekend. Yeah.
Definitely.
Yeah. And do you want to say something?
There were few questions as well that came like 5 to 10 minutes ago, and please post all your questions in the feedback forms.
Yeah, I'll look at an answer. I'll try my best to answer.
Yes.
Thank you, sir. Thank you, sir.
Okay. Okay.
Thank you.
Yeah.
Thanks.
Thank you very much, sir.
Okay. Thank you, everyone. Thank you so much.
Thank you, everyone.
Have a nice evening.
Thank you, Magesh, and thank you, Divyasri. Thank you.
Thank you, sir. Thank you, sir.
Thank you, sir.